I'm Callahan, and this project is using machine learning algorithms to predict the thermal stability of enzymes. The motivation behind this project is the fact that enzymes and proteins in general are used extensively in private industry and in research and potentially offer a lot of benefits in the future for things such as medicine, food production, other health-related tasks. And the difficulty is there's so many different proteins out there. Choosing which ones will have the right properties to actually research can be difficult. So rather than trying to exhaustively test all hundreds of thousands of different varieties of proteins, the goal here is, given the structure, can we make a machine learning algorithm that will reliably predict how much heat the protein can withstand, which suggests how usable it is in private industry. So the basic relevant biology here is the knowledge that proteins, enzymes specifically for this data set, are comprised of chains of amino acids. So there's about 20 different there's exactly 20 different amino acids, each represented by a capital letter. And these are chained together. In real life, they can have a wide variety of different shapes, but in the computer representation, they can be represented as a single vector, or a string rather. And given only this information, what we know about the amino acid structure, but not the physical structure that the amino acids imply, what can we infer about the chemical properties? So there's a history of proteins being investigated with machine learning algorithms and neural networks in general. The AlphaFold and AlphaFold 2 are some pretty well-known results where these large neural networks can very reliably predict certain types of protein structures. It's called protein folding. Essentially, what happens is, depending on the heat, depending on the environment they're in, proteins will either flatten out or sort of merge together in this tight bundle, and predicting the exact characteristics of that bundle and the properties that go along with it is a very important task that traditionally was just had to be done experimentally. But now, with these large neural networks, there's been a lot of progress made in deciphering what physical properties will be gained by different changes in the environment for these enzymes. Another less well-known result, but very relevant for some of the work that's being done here, is a different sort of neural network that comes out of a long line of neural networks, but it's by far the best performing, called deep DDG. And essentially, this neural network takes in strings representing amino acid chains, and given point mutations, given adjustments of the individual amino acids, how does that affect the chemical properties? And specifically, how does that affect the Gibbs energy, which is related to proteins flattening or folding? It's closely correlated with the structure that we're looking for here. So the actual target of these machine learning algorithms, what we're actually trying to infer from the data set, is the temperature of melting for specific enzymes. So we have a variety, about 30,000 different enzymes that have measured, they're related enzymes, they have a measured temperature of melting, and from that we want to take that information and be able to apply it to new enzymes where the structure is known, but the actual melting temperature is not. This is the reason I brought up DDG in the last slide, is because this is not exactly the same, but very closely related to the Gibbs free energy. So being able to properly manipulate these chains of amino acids and to understand their point mutations so that it can be fed through a neural network, like deep DDG is a big part of this process, and that contributes to one of the features that's going to be engineered later in this presentation. So the initial file is fairly simple. Basically, all you're given is all you're given in the primary CSV, the trained CSV, is the protein sequence, which is representing all the different amino acids in the chain, the pH, and the target variable, the temperature of melting. The pH has very little variation, but what's also provided for reference is results of previous neural networks. So results from AlphaFold on some relevant materials, results from deep DDG, which are available publicly. Those will come into play, but the primary factor here is the protein sequence. So the first step at addressing the lack of features in this data set is to take some really simple, sort of naively engineered features from the protein sequence. So these are not dependent on any domain knowledge about proteins, about enzymes. All these are simple estimations of what the sequence we're looking at is. So the easiest ones here are the length of the sequence, which varies widely, and the number of unique components of the sequence. So that's the number of different amino acids. Doesn't vary as much, but there does seem to be some significant difference for especially smaller protein sequences which have limited enzymes. There can be an increased ability to anticipate how they're going to behave. And so the initial distributions are a little skewed. The pH is almost exclusively around 7, which is the sort of even level. The sequence size and the number of unique amino acids in the chains are also skewed in different directions. So the first step here was to remove some of the outliers. Now, primarily this came in terms of reducing the outliers in sequence size, taking away the extremely large values. They're going to be harder to parse. They're likely going to be less representative of the data. This makes it a lot easier to work with. And there is also some pre-processing involved with the pH levels, even though not all of the sort of outlying pH levels are removed, because down the line there's some potential for outliers to have an impact, and that will be investigated later on. So one of the first feature engineering methods that came to mind, which is also independent of the physical or scientific nature of this data, is just trying to measure the distribution of different amino acids in these chains. So what percentage of a chain is comprised of one amino acid, how common are they, potentially gives information about the sort of chemical properties available. Now, this isn't completely deterministic. None of these give you an extremely strong understanding of the underlying phenomena, because in reality the amino acids are part of larger mutations that actually have the effect. But there is some correlation between the frequency of certain amino acid chains and the temperature sensitivity of the proteins they're constructing. The simple case from the last slide was just single amino acids, but it also makes sense to try and say what combinations, what permutations of amino acids have more of an effect. So the end goal here is to try and pick out the relevant components, the relevant sections, subsequences of these proteins to determine what mutations really have an effect on the structure and on the chemical properties. Now, to do that exhaustively is not possible. It's not computationally feasible. But to do a first few steps, for example, here is shown permutations of 2 amino acids together. I've also tried up to 4 amino acids together. That's a reasonable step to see if anything pops out as being especially correlated with the target variable. So that brings us to the metric that's being recommended for evaluation of these models, and it's somewhat atypical. Rather than using some common metrics such as RMSE, the goal here is to predict or to maximize the Spearman correlation between the sequences and features extracted from the sequence and the target variable. So the output from the machine learning and the target, it's not intended to be linear. What's important here is that the direction, the change in the temperature equilibrium for these enzymes, that it can be understood in terms of adding or deleting amino acids from the chain. So the sort of rank ordering of values that are coming out of the machine learning model are a higher priority than getting exact numbers. So with that in mind, with the idea of using correlation to select features or to evaluate the data set, I extended that to trying to extract to measuring what features are usable after extracting all these different permutations. So the vast majority of permutations that are measured when you're adding all these combinations together are not going to be useful. They're only going to bias the machine learning model. So what I did here is I took the correlation on the left and the p-value associated with the correlation, which is the likelihood that there is a relationship between two variables, between the output of the model or between the features and between the temperature. I used that to select a subset of all the generated features to actually pass to the model. And effectively, there's an interesting curve on the right here, which is to say the vast majority of the permutations that were measured are not really impactful, or doesn't seem to be directly impactful, directly related to the target. So eliminating a lot of those lower p-value combinations can allow for some improved results. And that's exactly what we see here. So just as an example, I varied this throughout fitting the model, but as an example, a minimum p-value of 1/2, so about a 50% chance that the two series are structurally related, and alternatively, the correlation of about 0.15. These are sort of the cutoffs that I use to narrow down the feature set for inputs to the model. And the important thing to understand about that is, again, it's very useful and it turned out significant results in the first few stages, but it's not a sustainable approach. So yes, it's fairly easy to compute for a large number of these things, the correlations and how likely they are to have an effect on predicting the target. However, it is computationally infeasible to do so, certainly for all 20 amino acid chains, all 20 different amino acids, and considering that chains can be not just permutations or unique combinations, but they're comprised of a wide variety of unique subsequences. Trying to evaluate these sequences entirely based on this sort of exhaustive approach is not feasible. So a quick sample here of what the data set looks like after taking all these extracted features. Still maintains the pH, the target variable, and the naive sort of estimates of the sequence size and the number of unique amino acids. However, now there's all these varieties of different variables between combinations of size 1 to 3. And these combinations that are chosen are in this data set, the top 50 that had the highest correlation or the highest likelihood of impact on the target variable. And so that approach is very simple and could be applied generally without any knowledge of biology. But here where the project is sort of being extended is in where real results are coming from are understanding the research that's in the field and how that can be leveraged to gain some more insightful, more precise features in the data set. So one of the first metrics that's useful here is based on the structure the enzymes were given. So essentially, in nature, there's some consistent structures for proteins, for enzymes, and then there's a lot of mutations from those common structures. The common structures are called wild types, and the mutated structures are called mutants. It's very easy to quantify the difference between the wild type and the mutant to see how far the mutant has deviated from the sort of parent structure, and that can be measured by a few different metrics, but the most common one, which is fairly simple as it's just a construct of adding and removing sequences from a chain, is called the Levenshtein distance. So this later step of feature extraction takes the Levenshtein distance, which is independent of the biological component, and adds a sort of something that's specific to biology here, which is an understanding of the cost of transitions between different amino acids. So the basic intuition here is that there might be amino acids that are fairly chemically similar or have similar properties when they're included in subsequences. And so shifting between them, a mutation from one to the other, may be less impactful than a mutation from a different set of amino acids. And basically what's happening here is empirically and theoretically, a sort of combination of the two, there's some results that have been published about the effective cost of making these moves, and that can be used in conjunction with the Levenshtein distance to give a more robust concept of distance between the parent, the wild type amino acids structure, and the lower level mutated structures. So using that, using the Levenshtein distance was one of the later components to try and fit a model. And throughout the process, I was fitting a variety of different models. What seemed to be the most consistently high performing is light GBM. There's a few different ensembles I used, primarily random forest and light GBM, as well as linear models, simple linear models for some of the simpler features. And it seemed pretty clear that these ensembles handled this sort of data, this sort of sparse data, much better than the linear models did. And fairly decent results, whereas linear models initially with the naive set of features were getting almost no predictive validity. These ensemble models quickly got to about a correlation of 1/2 with the target. And this was fit using a variety of hyperparameter fitting methods. Essentially, quite a few different hyperparameters can go into light GBM. Part of the customizability is why the model is so attractive. And I added different variables for that and ran it through a cross-validation version of a grid search algorithm to try and tease out what the best variables are to not overfit the model on the training set so that it can generalize well to extra data. So the last thing to mention here, which has sort of been alluded to already, is that there's a lot of more complex methods that I haven't really investigated. So something I have included in the notebook but haven't really utilized is importing some of this raw data output from other neural networks, that's deep DDG, AlphaFold, being able to use that data to make better predictions. Alternatively, there's a lot of simplifications that were made. The outlier process was sort of back of the napkin, and their transition matrix we used, the substitution matrix rather, there's a lot of different options available in the literature. That's a common version, but other possibilities exist that might give better results. And again, adjusting hyperparameters, adjusting default values, how you're filling in missing values, especially when you're querying databases that don't have information for all these protein sequences. Choosing what to include and what not to include there could have very significant results. And building on top of the feature selection that's already there, more robust feature selection methods could probably provide a lot better results in the long run.